All right, guys, picking up with Chapter 1, Part 2. We left off with our phylogenetic discussion. And so, picking up with the next slide, which I think is slide 44, right, we talk about the three domains that we currently use. All those are some debate. That's what's current. Um, and pretty much one, in my opinion, is kind of a go between. So, you got the domain bacteria. That is all your unicellular prokaryotes that live in all environments, including our normal flora, which are found on our skin and in our mouth and in our large intestine, right? But these are all unicellular and they all lack nuclei and defined intracellular compartments, as we discussed in part one. And then there's the domain archaea. They are prokaryotic, like their bacteria brethren, but they are also extremophiles, right? You find these in deep sea thermal vents and acidic ponds and places like that. But the key from an evolutionary standpoint is their membrane composition, the type of phospholipids that make up their outer membrane is very similar to eukaryotes. So it's thought that after, Leuca devolved into archaea and bacteria, that archaea at some point split and formed the eukaryotes, which we'll talk about the theory of endosymbiosis, right? That's going to play a big role in eukaryotic development and how they developed intracellular compartments. So three domains, right? As we talked about, the archaea, right? Only thing I really want you to remember is what I told you about their membranes and the fact that they are extremophiles. Right, and they are prokaryotic in nature, right? There aren't any pathogens in this group, so they're not really medically relevant. The domain bacteria, now these are the other prokaryotic domain. These are where you find your human pathogens, right? These are roughly subdivided when we get into micro into gram-positive and gram-negative organisms, depending on how they behave with a differential stain called the gram stain. Most of these guys are heterotrophic. In fact, they are called chemo organotrophs for the most part. There are a few chemolithotrophs, those that can synthesize inorganic nutrients. And like I said, the cyanobacteria is a big member, and these are actually photosynthetic bacteria from which it is thought that chloroplasts probably evolved. And we'll get into that when we talk about the theory of endosymbiosis. And so with the domain eukarya, it's much larger. There right now are 4 accepted kingdoms. The protists, right? A large portion of these are actually unicellular, but there are some multicellular members. These are mainly the aquatic algae protozoans, of which there are some pathogens. The famous, what do they tell you when you go to Mexico not to do? Don't drink the water, right? Protozoal infections. And then the domain of the kingdom fungi. Most of these are what we would call roughly the decomposers. The fancy term for that is they are saproprobic, which means they live off decaying or dead animal matter. So they're important as the recyclers. And then the 2 other obvious ones, the kingdom plantae and the kingdom animalia, right? Although more complex algae and some mosses, which could sometimes either fall into the protista or the kingdom fungi, are actually grouped in the kingdom plantae. And so, right, with bacteria and archaea, they're still developing kingdom designations. They hadn't got that far. And we talked about with eukarya, right, they have designated kingdoms, but the development of these supergroups, which are sometimes expected to fall in between domains and kingdoms, are actually being developed. And so if you wanted to look at an example of a eukaryotic supergroup, right, for example, the supergroup Excavata is the diplomonads and the euglenozoans, which, are from the collective group of protists, right? So it's just a way to further classify, right? And the thing about the supergroups is they're going to share similar characteristics. So it's just a new way to further subdivide, and the supergroup falls in between the domain and kingdom classifications. And so getting into a little discussion about what is science, right? Pretty much the ability to ask questions through observations and then design experiments to test those observations, right? With three basic principles. Events have natural causes, laws do not change over time or distance, and here's the hard one. Findings are value neutral, right? Meaning they're independent of bias. Right? A lot of times, guys will tell you, you can give me the data and I can make you look. I can argue it any way you want to, right? But true experiments are value neutral, right? We're not swaying them one way or the other. My mentor used to like to say, "It is what it is." Right now, go do it again. So when we talk about the scientific method, right, pretty much divided into 5 parts: the observation, right, something you see in the environment; a hypothesis, right? Something to explain the observation. And this is hopefully testable through a designed experiment. And based on the parameters of that experiment, you're going to have a prediction, right? Then you perform the experiment, which allows you to collect data, right, which is the currency of any experiment. You're going to run statistical analysis on that data, meaning are the differences, and basically with statistical analysis, are the differences due to something other than chance? Is the difference not just attributable to random variation? And I'm going to run out of room, but you get the idea. Okay. Then based off that analysis and that data, depending on what that data says, you're going to have results that lead hopefully to a conclusion. And so, right, with an observation, you may have several different hypotheses, which lead to predictions and experiments, which will allow you to reject an hypothesis. Every hypothesis is also going to have what I like to call a null hypothesis. So, for example, if I predict a certain species of bird will eat a certain type of seed, then obviously the null hypothesis would be that particular species of bird will not eat that type of seed. And then what would I do? I would design an experiment with different seed types, and I would expose that bird to those seed types and see which seed type he preferred, right? So that may or may not allow me to reject a hypothesis, and usually a rejected hypothesis leads to the formulation of a secondary hypothesis, right? New data sometimes leads to new questions. Or it allows you to modify the hypothesis. And so at what point your predictions become confirmed? Once your predictions are confirmed, pretty much that leads to a conclusion. And I am going to generate a short video for you guys. It's going to be a little bit separate from the lecture. We'll talk about inductive versus deductive reasoning and how they are used in the scientific community. And so pretty much as we defined an observation, scientists use their senses to gather information about a phenomena or natural event. And then the hypothesis is the tentative explanation for what was observed, right? We'll talk a little bit about history when we talk about the discovery of penicillin. And pretty much the idea of a hypothesis is developed through the use of inductive reasoning, right? And so with inductive reasoning, pretty much what you're looking at, or inductive reasoning, pretty much what you're doing is you're taking specific observations and then pretty much what you're doing is using those specific observations to come to a broad conclusion, right? So, an idea of inductive reasoning would be, my neighbor's cat hisses at me daily. At the pet store, all the cats hiss at me. Therefore, your conclusion, all cats probably hate me. Now, is that true? No, but it's based, or maybe not. It could be true, right? But it's based on all the data generated through those observations. And so, you know, another simplistic example of inductive reasoning might start an observation with all the cows I have ever seen are spotted. So you might think, well, all cows must be spotted. Now we know that's not actually the case, but given the available information, I could give you a pass for thinking that, right? But the next step in this logic might involve attempting to find more things which disprove the assertion that all cows are spotted, and you might do that by asking other people if they have seen cows which are not spotted. So inductive reasoning is commonly seen in the sciences when people want to make sense of a series of observations. Probably one of the most famous ones is when Isaac Newton used inductive reasoning to develop a theory of gravity. So, using observations, people can develop a theory to explain those observations and seek out disproof of that theory. As can be seen in the cow example above, one of the major flaws, or the cow example that I gave you, one of the major flaws with inductive reasoning is that it is dependent on observations. And when observations are incomplete, unsound reasoning may be formulated. Okay? A good example of this would be the idea of spontaneous generation, which we'll talk about a little bit later. So, an experiment is a series of procedures designed to test the hypothesis. Now, an experiment uses deductive reasoning to make an observation or to make a prediction or expected outcome. And so, with deductive reasoning, pretty much what you're doing is you're taking general ideas. We may not need this inductive reasoning video after all because we're talking about it here, I guess. I guess that's a good thing. You got general ideas leading to a specific conclusion. So quite different from inductive reasoning. You know, in practice, deductive reasoning, right? Really simple. A is B, C is A, therefore B is C. Another example. All muscles are made out of living tissue. All humans have muscles. Therefore, all humans are made out of living tissue. Right? So with deductive reasoning, one takes a general theory or idea, tests it, and moves through a sequence of ideas to arrive at a specific conclusion. So it's possible to arrive at an unsound result by using an initial premise which is false, right? But the goal of deductive reasoning is to arrive at a valid chain of reasoning in which each statement holds up to testing. So it's possible for deductive reasoning to be both valid and unsound. I'll give you an example. All men are mortal. John is a man. Therefore, John is modal. John is mortal. So all men are mortal. John is a man. Therefore, John is mortal. This is an example of valid deductive reasoning. On the other hand, here's an example of inductive reasoning. Inductive. Most men are right-handed. John is a man. Therefore, John must be right-handed. So the strength of that inductive argument depends on the percentage of left-handed people in the population. In any case, your conclusion may well end up being invalid because inductive reasoning does not guarantee validity of the conclusion. Another thing that they sometimes characterize deductive reasoning is what they call top-down logic. Now, what does that exactly mean, right? Whereas inductive reasoning is sometimes called bottom-up logic. So let's compare and contrast those statements, right? Deductive reasoning contrasts with inductive reasoning and generally starts with one or more general statements or premises to reach a logical conclusion. If the premises are true, the conclusion must be valid, right? Deductive reasoning is often used by scientists and mathematicians to prove their hypotheses. So with deductive reasoning, arguments may be valid or invalid, sound or unsound. It depends on if the logic is correct, right? However, valid arguments may be sound or unsound. If the premises used in the valid argument are true, then the argument is sound or is sound. Otherwise, it is unsound. Here, I'll give you an example. It's always better with the examples. All men have 10 fingers. John is a man. Therefore, John has 10 fingers. This is both logical and valid. right? However, the premise that all men have 10 fingers is incorrect because we know that some people are born with 11 fingers, polydactylism, right? Therefore, this is an unsound argument. Inductive reasoning or induction is reasoning from a specific case or cases and deriving a general rule. Now, this goes against the scientific method because it makes generalizations by observing patterns and drawing inferences that may be correct. So you get the idea about how those two types of reasonings have to play together, right? Inductive reasoning leads to the hypotheses, and hypotheses are tested by deductive reasoning. And so how do we set up these experiments? A test group, right, is exposed to the experimental variable, right? For example, if we're doing a medical study, right, those are the ones that quote unquote get the drug, right? And then the control group is the one that gets what we call the placebo, right? Instead of getting the drug, they get sugar pills, right? They're treated the same as the test group in all other aspects except for the experimental variable. Because you remember, good experiments are only going to contrast one variable. If you got multiple variables, how do you explain the effect, right? If the control and test group show the same results, then the hypothesis is not supported. And the data in this case are the results of the experiment, right? And we're going to examine those through statistical analysis because statistical analysis is pretty much going to say, if we do see the variation, is the variation significant, or can it be attributed to chance, just normal, variation in the process? And that's what we need to know, because if it is significant, then we need to look for the cause of that variation, and then hopefully the cause of that variation would be the difference in the experimental and control groups. And so we use the chi-square analysis, as we're going to go through later in the semester, right? We can use chi-square analysis to answer that question. Is it significant? And this generates probability values, right? And so what we're looking for in scientific studies are p-values that are less than 0.05 in our scientific analysis. Because what that means is, that means there's less than a 5% probability the results are random when we get those p-values, which means there's a 95% chance that those differences are significant. And so when we look at these graphs, a lot of times what you're going to see here are the error bars, right? And the error bars are the plus minus, meaning the variation in the numbers. And a lot of times these trials are presented as averages, right? Your data point are the averages of usually 3 trials, because most good experiments are done in triplicate. And then the error bars represent the variation in numbers, or in this case, the differences or the variation in 3 trials. So obviously, good experiments, we want these error bars to be small, right? If they're large, that means we got a lot of variance in the experiment, and it's not going to be easy to determine if it's statistically significant. And so the data are interpreted to determine whether the hypothesis is supported or not. If what we predict happens, obviously, the hypothesis is supported. If not, the hypothesis is rejected, meaning, and a lot of times they'll say, if the hypothesis is rejected, then the null hypothesis is accepted, which is the opposite of your hypothesis. And so, for example, in that last study, our prediction would be the drug would lead to an effect. The null hypothesis would the presence of the drug leads to no effect. Okay. Then we're going to report these findings, right? Most scientific journals are what we call peer-reviewed. And so what that means is, me being a bacteriologist, if I'm going to publish something in the Journal of Bacteriology, before they publish that article, there's going to be 3 guys that are also bacteriologists and work on similar things that I do. They're going to read my paper and say, yeah, he did this the way I would have done it. I buy those results. And then it goes on to publication. And so, you see a couple there, right? There's hundreds of different journals. It just depends on what field you're in. For example, in my field, I publish mostly in the Journal of Bacteriology or in Infection and Immunity because I worked on a pathogenic microbe, right? So those are the journals. If I'm an evolutionary biologist, I'm going to target magazines like or journals like Evolution, okay? And then eventually, right? Hopefully these hypotheses get tested over and over again, and the experiments keep generating the same results, and that pretty much leads to the development of a theory, right? When a hypothesis holds up and it's well supported, um, and also supported by a broad range of observations, experiments, and data. Theories can eventually lead to what we call a principle or law, right? These are pretty much accepted sets of theories, like the theory of gravity, right? That's pretty much is what it is at this point, and there's no serious changes to validity. So theories, right? They're pretty much accepted, but still tested and debated, and sometimes they are refuted. But once you get to the law set point, right, you're pretty much accepted, right? We accept it based on the information that we have at that time. And so the basic theories of biology are the cell theory, right? All organisms are composed of cells, and new cells only come from pre-existing cells, right? So once again, we get the process and mechanism, right? All organisms are composed of cells, and then where do those cells come from? They come from pre-existing cells. And then homeostasis, right? The internal environment of an organism stays relatively constant, that within a range that is protective of life, right? That's why this is going to vary according to the environment. And then evolution, all living organisms have a common ancestor, but each is adapted to a particular way of life, right? So the development of the cell theory, what an organism is made of, and the theory of evolution by natural selection, where do organisms come from, those are really the two big benchmarks of biology. And as we discussed, a theory, an explanation for a very general class of observations or phenomena, and they're divided into two parts. A pattern, which requires the observation, and then a mechanism, which is the process that explains the pattern. Okay? Theories established all organisms are cellular and change over time. And so microscopy was a big precursor to the cell theory, right? The fact that there were things that we couldn't see, and then the things that we could see had an inherent structure. And so Robert Hooke and Anton von Leeuwenhoek were two pioneers in the field of microscopy. Both of them designed their own microscopes. Hooke was given credit for coining the term cell, as he observes some tree bark and described the little compartments as similar to what you would see in a monastery, the monks' quarters, which were sometimes deemed cells. And then Leeuwenhoek is actually given credit for observing the first living molecules. He was looking at a couple of drops of pond water and described what he called animalcules, right, which were actually very large paramecium that you commonly find in pond water, right? So this was the pattern, right? All organisms are made of cells. And so you can see some shots. This is Hooke's microscope. He was part of the British Royal Society, so actually he had pretty good funding at the time, right? This is one of those that they have in the museum. But just to go shows you, you can do you can do more with less, right? This is Leeuwenhoek's microscope, and you can see very simplistic little structure. The drop of pond water would go here, right? And there's the lens apparatus embedded there, right? You got your adjustment for the type of specimen here. And so much rougher and not near as pretty as Hooke's microscope, but they both got the job done, right? And here's some of Leeuwenhoek's drawings of some observations that he made over time. Essentially, that guy had one great quality of a scientist. He was curious and he questioned things, right? So did a lot with little, okay? So Virchow in the late 1800s came up with the idea that all cells arise from pre-existing cells. The problem was you had to refute the process or the hypothesis of spontaneous generation, which was sometimes referred to as abiogenesis, which, life just spontaneously arises, which I guess it did happen once, at least with the Big Bang. But the idea that all life arose from non-living materials. And so, Virchow came up with this theory, but he didn't have any proof, right? And so we know with the complete cell theory, all organisms are composed of cells, and all cells come from pre-existing cells. This went right in the face of abiogenesis and the theory of spontaneous generation. And so it wasn't till Pasteur came along, Louis Pasteur, that we got conclusive proof that spontaneous generation was not a thing, okay? And so let's get to his experiment, right? He did what we call the classic, or you may have heard of it, the famous swan neck flask experiment. And the idea was he had 2 flasks of nutrient broth. These were boiled to sterilize them, right? One flask had an open or straight neck. The other one had this curved neck, which was compared to the shape of a swan's neck. And the idea is both had direct access to air, which the spontaneous generation guys argued was imperative for spontaneous generation to occur. And you guys know how this went, right? Dust gets into this guy, microbes are on the dust, the broth becomes contaminated, right? In this one, air still gets in, we got dust that collects here, right? And because the dust collects here and microbes don't get in, this stays sterile, right? And pretty much this rejects the hypothesis of spontaneous generation. So only one variable here, right? You guys can figure this out. What was the variable? The shape of the straight neck flask, right? It was either straight or it was swan neck. Everything else was kept the same, right? And that was the variable that was tested, okay? So, the spontaneous generation process hypothesis would be cells arise spontaneously from non-living materials. And then the all cells from cells hypothesis is cells are produced only when pre-existing cells grow and divide, right? So basically looking at these experiments, it provides proof for both, depending on how you want to look at it, okay? And so, looking at what the studies that we do, right, you're going to take a group of test subjects. You're going to divide them into groups, right? And then in this one, the control group, right, the placebo, that means no treatment. And in this particular example, you got 2 different antibiotics, right? And in this experiment, pretty much each subject was examined for the presence of ulcers, right? And then what they're testing is, the placebo group should have a certain number of ulcers because they receive no treatment. And then they're obviously looking for a reduction in those ulcers with the different antibiotics, right? So, maybe they discover that antibiotic B is a better treatment for ulcers than antibiotic A, and then antibiotic A is the one that's in current use, right? So they want to advocate, hey, we got a better antibiotic here, okay? So one of the control groups would include subjects with ulcers who are not treated, and then the 2 test groups would have subjects with ulcers that would get either A or B. And then you do an endoscopy, right, which is a procedure, that you can look down the throat and look at the stomach and the upper small intestine. That's actually a check for ulcers, right? And then looking at the number of ulcers, you generate statistics with and without those antibiotic treatments. And you make derivations or conclusions based on which antibiotic is better. And then hopefully that would be the one where the groups had the least amount of ulcers compared to the control group. And so, science today is totally changing because, as I like to say, science is totally related upon the environment. So as the environment changes, so does science, right? And technology is the application of scientific knowledge to the interest of humans, right? For example, obviously we'd be interested in that new drug that we just talked about that would reduce ulcers. And so some of the big changes that we're facing today are things like climate change, right? Changes in the normal cycle of the Earth's climate that are attributable to human activities, right? As we increase carbon emissions, that leads to the inability of some of that carbon to be absorbed, right? It gets stuck in the atmosphere, and and this causes a temperature increase, which is called global warming, which is collectively referred to as the greenhouse effect. And so obviously as environments get hotter, the environment changes, which may or may not have an effect on the species that live in that particular population, right? So when we talk about biodiversity, that's the total number and relative abundances or abundance of species, the variability of their genes, and the different ecosystems in which they live. And it's estimated that we may have on this planet approximately 8.7 million species, only approximately a quarter of which, 2.3 million, have been named and identified. And when a species disappears, that's what we call extinction, the death of the last member of a species or a larger classification category. And it's estimated that we lose hundreds of species every year due to human activities. For example, the reefs, right? For example, most of you, based in living in Wilmington, you're going to know about this kind of stuff. But these coral reefs, right? As the temperature increases, the coral becomes bleached and dies. And the problem with not only that happening is these coral reefs are habitats and protected places for hundreds of different species, and then also hundreds of different predators which live on those species. And so if those habitats die, then we may or may not lose those species in the process. Okay, the cutting down of the tropical rainforests. Not only are we cutting down a huge oxygen producing environment, right, but it's a huge ecosystem with, I think in the Amazon they estimated, I read a paper where they estimated there were 400 different species of tree frogs in the Amazon alone, right? So if you're removing that environment and those guys don't have time to adapt, those species may go extinct, right? And those are two really examples of habitats which are being threatened in our current environment. And we depend on these ecosystems, mainly for food, but also raw materials and some natural medicines. And so the planet's not getting any bigger. And so sometimes we're our own worst enemy. For example, the draining of wetlands in the Mississippi and Ohio rivers due to increasing populations and land development has led to increased flooding in those areas, which leads to ruined farmland, which then leads to decreased food production, which leads to an increase in inflation, which also increases the amount of hungry people in the United States. The destruction of the South American rainforest, not only the loss of biodiversity, which we talked about, but because this is not done in an ecologically sound way, meaning for everything you harvest, you replant, eventually leads to a decreased availability of lumber, which leads to increased costs. Now, this constant foraging into new environments has also led to an emergence of what I like to call a huge category of zoonotic diseases. And these are diseases acquired from animals. And for hundreds of years, we hadn't had close contact with some of these animals. But as we continue to populate out and clear land and move into new environments, we infringe upon their habitats and become increasingly exposed to them. In fact, they think that's what happened with the COVID virus, depending on your politics, right? And then also the use of technology. Legionnaire's disease was an extremely rare disease, but now we see periodic outbreaks because that guy is really good at living in stagnant water. And there's plenty of stagnant water in air conditioning systems, right? So that's why we've seen an increase in the outbreak of what used to be a fairly rare disease. In fact, named after a particular type of soldier that was the only one that ever got it. And then globalization, right? As we continue to clear out habitats, and then as we expose ourselves to animals and expose other animals to different animals, we see pathogens mutating and then jumping hosts, right? For example, avian flu. It's thought that avian flu can sometimes recombine or jump to swine flu, and then that leads to epidemics in the human population. So these are all things that we're dealing with in the new world, and it's things that your generation is really, really going to be left holding the bag with if our current generation doesn't do something to start combating these temperature changes. So that's pretty much an introduction, right? We'll get these produced, and get them up for you. And make sure to check Blackboard for announcements on due dates and assignments. All right.